I'm going to present you the Gao team work on RGD-dependent response to shear stress in the article published in Biotechnology Progress in 2007. RGD stands for arginine, glycine, and aspartic acid. Proteins with RGD domain binds to specific ligands, as illustrated here with human integrin. RGD domains are known for inducing stress response, but also for their various biomedical applications, such as tumor imaging or drug delivery tools. The work carried out by the Gao team is key in understanding shear stress in plants. They uncover how the mechanisms involving RGD peptides we are familiar with in animals can also be found in plants. In this presentation, I will first give you the context, where you see that shear stress is a key problem for the production of therapeutic agents with bioreactors. Then, the analysis of Gao team work will show that RGD peptides inhibits all of the key elements in the shear pathway by interacting with at least 4 families of proteins. And finally, we will analyze the results published since the Gao team work and see a protein called AT14A could possibly be of great importance. Numerous therapeutic agents like anti-cancer drugs such as Taxol have been extracted from plants like Taxus cuspidata, and these taxus can be grown in bioreactors. Bioreactors have been developed to increase the yield of culture cells and the production of drug. Indeed, bioreactors are a high-scale cell culture tool, thanks particularly to the continuous turnover of the medium. This turnover is mediated by an agitation system, which aims to maintain the culture medium homogeneous. The major issue regarding this technique is the shear stress. Basically, we have contacts between cells with different speeds and trajectories. These differences can be noticed also on cellular scale. Though shear stress and mechanical constraints apply on cells, it is worth to notice that an excessive deformation will lead to reduced viability or even the cell apoptosis. Therefore, it will decrease the production yield of the therapeutic agent, explaining our focus on shear stress in plants. Here is the state of the art about shear stress at article time, meaning the scheme is a simplified view of the current knowledge on this field. Lectins and associated proteins will act as the first elements in mechanoperception. Here, the most important point is the role of a putative integrin-like protein. This putative integrin-like protein will activate the MAP kinase cascade, which in turn will activate reactor oxygen species, phenolic concentrations, and alkylization. There is another pathway on which I'll come back later, with associated kinase and EGF. We will now focus on the putative integrin-mediated response. Integrin, MAP kinase, ROS, phenols, and alkylization. Let's sum up. We have seen what are RGD, what is shear stress, and why is it important. Now we want to know how the Gao team article has characterized RGD-dependent response to stress. The following experiments will do the competitive inhibition. We have 2 actors, natural proteins with RGD domains inducing stress response, and synthetic RGD peptides. Peptides and proteins links to the same receptors, but peptides doesn't activate the response. So please keep in mind we will observe RGD peptide effects, meaning the opposite of RGD dependent response. RGD peptide will be compared to RGE peptide because of the structural similarity. For all the following experiments, we look at 4 different setups we will represent with the following color code. First one, in blue, absence of stress will be the control situation. Second, shear stress only in red, and third, shear stress plus RGE in orange gives similar results. Last situation, shear stress plus RGD in green give an intermediate response. First, study alkylization, since it is a classical response to stress. Here we have the evolution of the pH with time. Without stress, which I repeat is the control test, we observe a constant level of pH around 5.7. With continuous stress only, we have a quick and significant increase of pH, which stabilizes around the near neutral pH. With shear stress and RGD peptide, we have a moderate increase of pH, stabilizing around 5.9, whereas stress plus RGE give an important increase of pH, similar to stress-only situation. RGD response is significantly less than the two situations with shear stress and also significantly more than the situation without stress. These results show that RGD peptide is responsible for partial inhibition of the alkalization response. Then, the authors focused on hydrogen peroxide production. We have pictures of representative cells in each condition. On the left, you have contrast photos of Taxus cuspidata cells, and on the right, the corresponding images obtained by fluorescence. Rhodamine was used as a probe to highlight hydrogen peroxide concentration. Rhodamine fluorescent signal stands for hydrogen peroxide production. Without stress, we have a low level of hydrogen peroxide. With shear stress, we have an important concentration of hydrogen peroxide, and with shear stress plus RGD, we have an intermediate production. These results show that addition of RGD inhibits the production of hydrogen peroxide. This is convergent with the previous result of inhibition of extracellular alkalization. Altogether, we can conclude that RGD peptides are involved in the alkalization response. After that, they also experimented on the effect of RGD on other reactive oxygen species. They measured absorbance to obtain gross concentration at the end of stress. Without stress, we observe a basal level of hydrogen oxygen species. With stress, with or without RGE, we have an important increase in mass production. And with addition of RGD, we observe an intermediate response. Therefore, the inhibition observed of hydrogen peroxide can be generalized to other reactive oxygen species. Next response, phenol production is similar. Without stress, we have a basal level of phenols. With stress, with or without RGE, moderate increase, important increase, and with stress plus RGD, intermediate response. MAP kinase activity is tested with the Western blot against phosphorylated MAP kinases. This Western blot detects the 44 and 46 chylodietone MAP kinase, which mediates response to various types of stress. In this experiment, in the first time, cells are submitted to a moderate level of stress, and in the second time, they are allowed to rest. The stress is induced by the rotation rate. Three conditions are tested with this protocol. The first one tests the effect of the source shear stress. Observe a quick and significant increase in MAP kinase activity for both the 44 and the 46 chylodietone MAP kinase. Second situation, RGD peptide plus shear stress. We observe almost no phosphorylation, meaning no activity of the two MAP kinase. Third condition tests stress plus RGE. We observe a quick and significant increase of activity comparable to stress-only situation. Thus, we can conclude that RGD inhibits MAP kinase activity, and this inhibition is highly RGD specific. So, RGD inhibits all of the key elements in the putative integrin response pathway: MAP kinase, ROS, phenols, and alkylation. The next step is to identify the partners interacting with RGD. Therefore, the authors use trypsin to fragment these partners when binding to RGD. Then, they analyze and sequence the fragments by mass spectroscopy. The results lead to four families of proteins. First one, von Willebrand factor A dominant proteins are implied in cell adhesion and the formation of multi-protein complex. Second one is collagen, which played an important role to maintain the structural cell integrity. Then, thyroid protein-like. And last one, integrin alpha chain, which was already supposed to play a role in stress response. Now, let's sum up the results of the article. First, RGD peptides inhibit stress response key elements. It means that proteins with RGD dominance will activate this response. Remember the beginning of my speech. So proteins with RGD domains activate MAP kinases, akinization, ROS, and phenol production. Secondly, RGD partners have been identified. There are boundary-bound proteins, collagens, cell wall proteins, and integrins. And thirdly, we've noticed that inhibition were always partial, which lead to the hypothesis another shear response pathway. Now, let's have a look at the results published for the last 6 years.